Hello, assalamu alaikum. Today, and we want to continue in chapter 18 after we talked about gluconeogenesis, which is the net synthesis of glucose starting from small non-carbohydrate precursors such as lactate, pyruvate, glycerol, and alanine. We said in this pathway, we must bypass the three irreversible steps in glycolysis. Now, the two reactions in which gluconeogenesis differs from glycolysis are ones in which a phosphate ester bond to a sugar hydroxyl group is hydrolyzed. The reaction 1, hydrolysis of fructose 1,6-bisphosphate to produce fructose 6-phosphate and phosphate ion. Notice that this reaction is exergonic, catalyzed by fructose 1,6-bisphosphatase, an allosteric enzyme strongly inhibited by AMP but stimulated by ATP. And also the enzyme is inhibited by this sugar phosphate called fructose 2,6-bisphosphate. And control point in the pathway because of allosteric regulation. This is the reaction, as you can see, hydrolysis of fructose 1,6-bisphosphate to fructose 6-phosphate catalyzed by the enzyme fructose 1,6-bisphosphatase. Now the second reaction is the hydrolysis of glucose 6-phosphate to glucose and phosphate ion. Notice again delta G naught prime is negative, which means it is also an exergonic reaction. The enzyme that catalyzes this is called glucose 6-phosphatase. Remember, this enzyme is found only in the liver, and it occurs in the endoplasmic reticulum. So here, this is an example of an interesting pathway, meaning the gluconeogenesis, which occurs in three cellular locations: mitochondria, cytosol, and endoplasmic reticulum. This is the reaction, hydrolysis of glucose 6-phosphate to glucose, catalyzed by this enzyme, glucose 6-phosphatase, which requires Mg+2 as a metal ion cofactor. Now, control of carbohydrate metabolism. Now, different enzymes catalyze opposing reaction in glycolysis and gluconeogenesis pathways. If you look at this figure, which summarizes the various control points in both glycolysis and gluconeogenesis, let's look at them. Notice here on the left, this is regulation of glycolysis. Reaction number 1, catalyzed by hexokinase, is inhibited by its product glucose 6-phosphate. Reaction number 3, catalyzed by phosphofructokinase, which is inhibited by both ATP and citrate, but activated by AMP and fructose 2,6-bisphosphate. On the other side, regulation of gluconeogenesis, you see these two allosteric effectors themselves have the opposite effect on the enzyme called fructose 1,6-bisphosphatase. AMP inhibitor, fructose 2,6-bisphosphate inhibitor. And this is logic, because these two enzymes cannot be active at the same time because they catalyze opposing pathways, breakdown of glucose and synthesis of glucose. Then reaction number 10 in glycolysis, catalyzed by pyruvate kinase. You see it is activated by fructose 1,6-bisphosphate, but it is inhibited by the following: acetyl-CoA, ATP, alanine, cyclic AMP-dependent phosphorylation. However, the first enzyme in gluconeogenesis, pyruvate carboxylase, is allosterically activated by acetyl-CoA. So if you memorize this, you will be in good shape. All the control is here. All the control is here. And again, here we summarize in gluconeogenesis, hydrolysis of fructose 1,6-bisphosphate to fructose 6 is catalyzed by fructose 1,6-bisphosphatase. This enzyme is subject to allosteric inhibition by fructose 2,6-bisphosphate and AMP. In glycolysis, PFK catalyzes the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. This enzyme is activated by fructose 2,6-bisphosphate and the AMP as inhibited by ATP and citrate. Notice all these allosteric effectors are in the previous figure that we talked about. So go back to that figure and you will be. Now we want to talk about what is the role of this sugar phosphate called fructose 2,6-bisphosphate. This is the structure of this sugar phosphate. They found that the intracellular level of this fructose 2,6-bisphosphate is a key point here, where high concentration stimulates glycolysis and low concentration of it stimulates gluconeogenesis. Now, the concentration of this sugar phosphate in a cell depends on a balance between the synthesis catalyzed by enzyme called phosphofructokinase 2 and its breakdown catalyzed by fructose 2,6-bisphosphatase 2. As we will see again, now the enzymes that control the formation and breakdown of this sugar phosphate, here we have breakdown and here we have synthesis. The enzymes that control the formation and breakdown of fructose 2,6-bisphosphate are themselves controlled by dephosphorylation mechanism similar to the one we saw for glycogen phosphorylase and synthase. Now, both enzyme activity are located on the same protein, which is a dimer of 100 kilodalton molecular mass. Now here, if we look here, this is our protein kinase. This is the protein kinase. This is the protein dimer in which the two enzyme activities are located. Now, phosphorylation of the dimeric protein leads to an increase in activity of fructose 2,6-bisphosphatase, as you can see here, وبالتالي breaking down the sugar phosphate. And there is a decrease in its concentration, thus stimulating gluconeogenesis. However, the opposite process, dephosphorylation of the dimeric protein, leads to an increase in the activity of phosphofructokinase 2 and an increase in the concentration. Here we have synthesis of the sugar phosphate. And this, of course, lead to an increase in the concentration of this sugar phosphate eventually stimulating glycolysis. So again, phosphorylation activate this enzyme and inhibit this enzyme. Again, these two enzymes, one in glycolysis and one in gluconeogenesis, they cannot be active at the same time. Otherwise, there will be waste of energy. Now, this sugar phosphate, as we said, activate phosphofructokinase 1 and inhibit fructose 1,6-bisphosphatase. And this is the role of this sugar phosphate in controlling both processes. Now, the net result is similar to the control of glycogen synthesis and breakdown, the one we saw at the beginning of this chapter. Now, if we look at this figure, which shows us the allosteric effects in the control of carbohydrate metabolism, relative activity of the enzyme called fructose 1,6-bisphosphatase in the presence of various concentration of its inhibitor, fructose 2,6-bisphosphate, as you can see here. The inhibitor works by itself in lowering the activity of the enzyme. However, its effect greatly increased by the presence of the allosteric inhibitor called AMP. Now, they assay the reaction in the presence of 10 micromolar of AMP and 25 micromolar of AMP. And you can see there is a more decrease in the activity as the concentration increases. The activity was measured in the presence of 10 micromolar fructose 1,6-bisphosphate, which is the substrate. Now, the breakdown and synthesis of glycogen exhibit similar reciprocal regulation. So glycogen synthase and phosphorylase are controlled by two types of control: covalent modification, which is phosphorylation, as well as allosteric control by allosteric effectors such as glucose, ATP, AMP, and glucose 6-phosphate. Now, the enzyme system is responsible for the modification of both the synthase and the phosphorylase. We saw that phosphorylation of the phosphorylase activates, whereas phosphorylation of the synthase inactivates. Again, these two processes cannot work in the same way, at the same time. Also, hormonal control is of paramount importance in glycogen synthesis and breakdown. Now, an enzymatic cascade triggers the activation of glycogen phosphorylase, as we will see in the next figure. Here, this figure shows us the hormone activated enzymatic cascade that leads to the activation of glycogen phosphorylase. As a result of stress hormone called epinephrine may be released, and it will activate an enzyme called adenyl cyclase, which will convert ATP to cyclic AMP. This cyclic AMP will activate a cyclic AMP-dependent protein kinase, which in turn will activate a phosphorylase kinase, which in turn will activate the glycogen phosphorylase by phosphorylating it. So form B is converted to form A. The phosphorylated form is the active one. This is called cascade. All of these are called cascade effects. Now in the book, we have a couple of pages are omitted. Don't worry about because I did not mention them in the slides. So whatever in the slide is the one that is required from you. Now, glycogen metabolism in body organs. How do different organs share carbohydrate metabolism? Now, using combination of these control mechanisms, an organism can set up a division of labor among tissues and organs to maintain control of carbohydrate metabolism, and a clear example is found in the so-called the Kury cycle. The Kury cycle is named for Gertie and Carl Kury, who first described it. Now, there is cycling of glucose, then involves cycling of glucose due to glycolysis in muscle and gluconeogenesis in liver. So the Kury cycle operates between liver and glycogen between liver and muscle. In muscle, we have glycolysis, glucose is consumed, and in the liver, we have gluconeogenesis, glucose is produced. Now, glycolysis in fast-twitch skeletal muscle produces lactate in conditions of oxygen depth. Gluconeogenesis recycles the lactate to glucose after it is oxidized to pyruvate. Of course, this occurs in the liver. And then glucose is transported back to skeletal muscle by the blood. Now, the buildup of lactate contributes to the aches, muscular aches that follow strenuous exercise. Now, gluconeogenesis recycles the lactate that is produced. This process occurs to a great extent in the liver after the lactate is transported there by the blood. Glucose produced in the liver is transported back to skeletal muscle by the blood, where it becomes an energy store for the next burst of exercise. Now, this is the main reason why athletes receive post-event massages and that they always cool down after the event. Cooling down keeps the blood flowing through the muscles and allow the lactate and other acids to leave the cells and enter the blood. So massages increase this movement from the cell to the blood. Now, if we look at the so-called the Kury cycle, you see here a summary of the Kury cycle, which operates between liver and muscle. Here in the muscle, we have severe exercise as a result of exercise, or glucose is converted all the way to lactate with the production of NAD2 ATP. Then lactate takes its time, it goes out to the blood, and then the blood transports lactate to the liver, where lactate now is the precursor for gluconeogenesis, converted to pyruvate, and all the way to glucose as we talked in gluconeogenesis. It consumes how many? 6 NTP. What is this N? N for nitrogen base. You remember this process consumed 4 ATP plus 2 GTP. So this N could be adenine or guanine. And this N down is only adenine. So this is 2 ATP. Now, after glucose is synthesized in the liver, it goes all the way through the blood back to the muscle, where it is used for another exercise, and so on. Notice here, in the muscle, we have low NAD plus to NADH ratio. And in the liver, we have high NAD plus to NADH ratio. Now, NAD plus, that is produced in glycolysis. Oh, this is NADH produced in the glycolysis is reused in gluconeogenesis. And there is the recycling of NAD+. Here it's a product, here it's a reactant, and so on. So this is, now if we look at the if we look at the two processes together, we notice that the glycolysis, one glucose is converted to 2-pyruvate plus 2 NADH plus a net of 2 ATP. In gluconeogenesis, 2-pyruvate plus 2 NADH plus 4 ATP plus 2 GTP synthesize 1 glucose. Now, if we add them together, we have this overall 2 ATP plus 2 GTP are hydrolyzed. So, the hydrolysis of both ATP and GTP is the price of increased simultaneous control of the two opposing processes. So if the cell controls both processes at the same time, it must expend energy, and that is the hydrolysis of 2 ATP plus 2 GTP. Now the last section of this chapter, 18.4, glucose is sometimes diverted through the pentose phosphate pathway, which is an alternative to glycolysis and differs from it in several ways. In glycolysis, one of our most important concerns was the production of ATP. However, in this pathway, the production of ATP is not important per se. As the name indicates, 5 carbon sugars, including ribose, are produced from glucose. So glucose is converted to ribose. Ribose and its derivative, hadol, they are important for nucleic acids synthesis. Another important point in the pathway is the production of NADPH, a compound that differs from NADH by having an extra inorganic phosphate. And you can see this here. This is the structure of NADPH, and you see that this is the only extra point, extra inorganic phosphate, NADH and NADPH. This is the difference between them, the presence of this. Now, the more important difference is the way these two coenzymes function. Namely, NADH is produced in the oxidative reaction that gives rise to ATP, whereas NADPH is a reducing agent in biosynthesis, which by its very nature is a reductive process. And we will see this in chapter 2, how NADPH is used in the reductive biosynthesis of lipids. Now, this pathway begins with a series of oxidation reduction reactions to produce NADPH and 5 carbon sugars, and is followed by non-oxidative reshuffling of the carbon skeletons of the sugars involved. So let's see the first reactions called oxidative reactions. So we have reaction 1, glucose 6-phosphate is oxidized to 6-phosphogluconate, catalyzed by enzyme called glucose 6-phosphate dehydrogenase, results in the production of NADPH, as we will see. Reaction 2, oxidative decarboxylation, where 6-phosphogluconate molecule loses its carboxyl group, which is released at CO2, catalyzed by 6-phosphogluconate dehydrogenase, results in the production of the 5-carbon ketose called ribulose 5-phosphate. Let's see this here in the pathway. You see here, this is the first part, the oxidative part. This is the first part, the so-called oxidative part. We start with the glucose 6-phosphate in the presence of enzyme called glucose 6-phosphate dehydrogenase. The oxidizing agent here is NADP plus, reduced to NADPH, and we form 6-phosphogluconate. So the aldehyde here is oxidized to carboxyl group with the production of 1 NADPH. Then a second oxidation reaction occurs where 6-phosphogluconate is oxidatively decarboxylated to ribulose 5-phosphate with the production of a second NADPH. Notice here that 1 carbon, the COO minus was lost as CO2 in the reaction. So we call this oxidative decarboxylation reaction. So in summary, this part, the oxidative part, results in the production of 2 NADPH, which are used in the reductive anabolic pathways, and ribulose 5-phosphate. Ribulose 5-phosphate will be used in the ribulose 5-phosphate will be used in the second reaction is called non-oxidative. In the non-oxidative pathway here, ribulose 5-phosphate undergo two changes. One molecule isomerizes to form ribose 5-phosphate. Notice here, this is the one that is important in nucleic acid biosynthesis. And the second molecule is converted to xylulose 5-phosphate by an epimerase. Here, look at the carbon number 1, 2, 3 here. Carbon 3 OH is to the right. Here OH is to the left. So we call this inversion of configuration to form xylulose. Now we have ribose 5-phosphate and xylulose 5-phosphate. Then a reshuffling of carbon atoms occur in the following reactions catalyzed by two enzymes. One is called transketolase and the other is called transalkylase. Now, transketolase transfer a 2 carbon unit. Transketolase transfer a 2 carbon unit. And here we transfer 2, we add them here to get a compound called pseudoheptulase 7-phosphate. This is 7 carbon. And what remains from the 5 carbon, 3 carbon, called glyceraldehyde 3-phosphate. Then a second enzyme called the transaldolase will catalyze a transfer of 3 carbon units. And we take 3 from the 7, add them on the 3, we get a 6 carbon called fructose 6-phosphate. And what remains from the 7 carbon, 4 carbon called erythrose 4-phosphate. Okay. Now, we get another molecule of xylulose 5-phosphate plus erythrose 4-phosphate. And the same first reaction is repeated here, transketolase, a 2 carbon transfer. We take 2 from xylulose, add them here, we get a second molecule of fructose 6-phosphate. And what remains from the 3 carbons called glyceraldehyde 3-phosphate. So as you can see here, in the pentose phosphate pathway, glucose 6-phosphate can be converted to fructose 6-phosphate and glyceraldehyde 3-phosphate, as you see, by means other than the glycolytic pathway. For this reason, the pentose phosphate pathway is also called the hexose monophosphate shunt, and this name is used in some texts. A major feature of the pentose phosphate pathway is the production of ribose 5-phosphate and ribose 5-phosphate and NADPH